Bismillahirrahmanirrahim. Today we'll discuss the humoral immunity that is a hot topic nowadays, especially in this current pandemic of the COVID-19. When we talk about the humoral immunity, it is basically the antibody-mediated immunity, and this antibody-mediated immunity it is primarily directed against the exotoxin-mediated diseases, and these exotoxin-mediated diseases they are mainly the diphtheria and the tetanus. Tetanus is very common in our society, especially when we look at the various infants and the neonates. It is common. You'll come across many patients in the neonatal ICU, neonatal intensive care units, who are suffering from the this tetanus. This humoral immunity it plays a very important role in the various infections related to the capsular bacteria, and those capsular bacteria, as you already know, there are three very important capsular bacteria which include the Haemophilus influenzae, pneumococcus, and the meningococcus. And this humoral immunity it is also directed against certain viral infections. When we talk about the humoral immunity, it is divided into the primary response and the secondary response. And both of these responses, the primary and the secondary, they need to be elaborated in detail. They need to be studied in detail. When IgM will be formed, when IgG will be formed, when the level of these immunoglobulins they will be on the peak. All these things they need to be elaborated in this humoral immunity. So first of all, uh, before starting this primary response and the secondary response, I'll discuss a term that is called as lag period. This lag period it is usually the time period between the exposure of the antigen and after the exposure of the antigen. It is the time taken, the number of the days for the development of the immunoglobulins. So this is the lag period. For example, if a person is exposed to antigen on day 0 and the antibodies it develops on day 7, it means there is a lag, there is a delay in the development of those antibodies. There is a delay of 7 days in the development of those antibodies. So this is a lag period. When we talk about the primary response, the primary response it occurs when we encounter an antigen on the first time. Like nowadays, most of us we are encountering the coronavirus, the COVID antigen. So this is the primary response. The antibodies they are detected after a longer lag period, and that lag period in case of primary response it is usually 7 to 10 days, but it can be longer than 7 to 10 days. So 7 to 10 days they are must after the primary exposure to the antigen. After 7 to 10 days, then the immunoglobulin levels will start rising. The initial immunoglobulin molecule that is formed in the primary response is the immunoglobulin M, and this immunoglobulin M, after about a week or so, or you can say at about 14 to 15 days after the primary exposure, its level will start declining. When the level of immunoglobulin M molecule it starts declining, the level of immunoglobulin G it starts increasing, and it increases more than the level of immunoglobulin M. So the things to note about the primary response is a longer lag period of 7 to 10 days, level of immunoglobulin M that rises after 7 days or 10 days, and the level of immunoglobulin G that starts increasing when the level of immunoglobulin M that start decreasing. And remember, this immunoglobulin G its level it rises more than that of immunoglobulin M molecule. On the other part of this picture you can see the secondary response. Secondary response it occurs when we are exposed to the same antigen or some different antigen having same structure, same chemical formula, or you can say the antigen having the same molecular organization. So when we encounter the same antigen second time, maybe after years or months later, in the lag period in this case, that will be small. The lag period, it will be usually 3 to 5 days as compared to the lag period in the primary response. Now here you will remember the memory cells, the memory T cells, the memory B cells. The body, it has the memory cells which remember that this antigen was once encountered by the immune cells. So immediately after about 3 to 5 days, the level of immunoglobulins, they will start increasing. So the important difference from the primary response, the lag period is longer in case of primary response, while it is shorter in case of secondary response. The lag period is usually 3 to 5 days in secondary response or secondary exposure to the same antigen, while in case of primary exposure to the same antigen, it is about 7 to 10 days. The second important thing is, after about 3 to 5 days, the level of immunoglobulin M, it will start increasing and then gradually declines towards the end of second week. Now here the important point is, when the level of immunoglobulin M, it starts declining, just like the primary response, the level of immunoglobulin G, it starts increasing. But here the amount of immunoglobulin G or the level of immunoglobulin G, it will be much larger, it will be much higher as compared to that of the primary response, and it will remain for a longer period as compared to the primary response, as it is quite evident by this graph. If you look at this graph and compare the level of immunoglobulin G molecules, immunoglobulin G, in both the primary and secondary response, the level of IgG in secondary response is much higher as compared to that of the primary response, while the level of immunoglobulin M, that is somewhat the similar. So this is the important thing to note in the primary and the secondary response. So along with this primary and secondary response, two other things or two other terms, they need to be studied. One is somatic hypermutation, second, that is not mentioned here, that is the affinity maturation. So this somatic hypermutation and the affinity maturation, they are closely related to each other, and they are part and parcel of this humoral immunity. So what is somatic hypermutation? Let's suppose if we encounter an antigen, initially antibodies are formed after a longer lag period. Afterwards, years or months later, we again encountered the same antigen, and at that time the antibodies, they are formed quite earlier, and they behave in a more different and organized manner against the same antigen. So in the second exposure, the antibody binding to the same antigen, it becomes more firm and it improves, because there occurs some mutation in the DNA that encodes the antigen binding sites, and this process, it is called as somatic hypermutation. So on the second exposure to the same antigen, the antibodies, they bind to the same antigen more firmly, and the improvement in this antigen antibody binding, it occurs due to some mutations in the DNA that encode the antigenic binding sites, and this process is called as somatic hypermutation. The second important term that is related to this somatic hypermutation, that is the affinity maturation. So what happens in affinity maturation? Few mutations, they result in insertion or involvement of some different amino acids in the hypervaluable region, and these amino acids, these different amino acids, they result in some good antigen antibody bindings. The antigen and antibody binding, it occur more strongly due to the insertion of these new good amino acids, and as a result, the plasma cells, with these improved hypervariable region, they are more strongly selected by the antigens, and therefore they will be those plasma cells, they will be the main pool of the plasma cell that will be selected by the antigen, and these plasma cells will ultimately resulting in the secretion or the formation of the various antibodies. So this somatic hypermutation, that is quite evident. Some mutation occurs in the DNA and resulting in improved antigen and antibody binding. And what is affinity maturation? Some mutation occurs which results in insertion of some different amino acids in the hypervariable region, and these different amino acids, different amino acids, they result in improved antigen and antibody binding. And these cells, these plasma cells having these different amino acids, they are selected by the antigens, and ultimately they will be activated, resulting in the formation of the immunoglobulins. So this is the affinity maturation. So these two terms, they are important. They can be asked in the viva, they can be asked in as a small part of a question in the form of SEQ. Another important thing is the function of the antibodies. Briefly speaking, one or two important functions, they are just they can be asked unless the function of the antibodies. So the main important function is it helps in the neutralization of the various toxins. These antibodies, it helps in the neutralization of the viruses, and it also causes the opsonization of the bacteria and other microorganisms. So these are the 2 or 3 important functions of the antibodies. You just have to enlist the functions: neutralization of the toxins, neutralization of the viruses, opsonization. That is a process by which the microorganism, they are more easily digested by the phagocytic cells. Third important thing that is present in this slide is the tests for evaluation. How you will evaluate the humoral immunity. This is usually done by a process that is called as the nephelometry. We measure the level of the important immunoglobulin molecules, especially the IgG and IgM in the patient's serum, and in some cases we also measure the level of immunoglobulin A molecule. So along with this nephelometry, another important method, that is the immunoelectrophoresis, that can also be helpful in measuring or in evaluation of the humoral immunity. So the main antibodies that are used to measure, or that are routinely used for the evaluation of the humoral immunity, they are IgG, IgM, and in few cases we also measure the levels of immunoglobulin A molecule. So this was a brief lecture about this humoral immunity. I hope you people must have understand this lecture. If there are some points or something that you people do not understand, please raise those points. I'll definitely try to answer these questions. As you know that I am extremely busy these days, so this is quite difficult for me to make a lecture, to upload a lecture, but inshallah ta'ala I'll definitely try to upload the lectures and to finish this topic of immunology. So till the next lecture, stay blessed and Allah Hafiz.